reckon. I'll just share my screen. Yeah, I'll share my whole screen. Okay, and you guys let me know if you can see that. Bring up the markdown. Can everyone see my RStudio?
Yes.
Yes, you can.
Great, thanks. All right, so for those of you who don't know me, my name is Emily. I'm a PhD student at JCU. I work with GPS telemetry with sea turtles, and I'm trying to understand questions about their fine scale movement behavior, also their habitat use and resource requirements, and the risk of their interactions with detrimental interactions with humans. Why might we be interested in studying animal movement? Well, the kinds of tracking devices we use in animal telemetry fields allow us to collect often really fine scale information on animals in parts of their life or in places which might be difficult to access by traditional survey methods. So that's one huge advantage. It can give us information on their distribution, their behavior, understand why they go where they go, and their interactions with other organisms. And often animal movement data is a really sort of charismatic tool, not only for providing this fine scale information, but also engaging managers with strategies for conservation of threatened species or control in the case of pest species. I just want to point your attention towards a little resource, which I'm not sure if anyone will be familiar with, but it's MoveBank. So if we follow this link to MoveBank, it will take us to the homepage of this website, hopefully. Yeah, there we go. So MoveBank is an online platform that has kind of come about as an initiative to get people to take their tracking data and put it online and make it accessible to other people. This is really, really cool, really great, because it means that researchers have access to much larger scales of data. We can start looking at more global questions and answering questions with larger sample sizes. Often a limitation of our tracking is that we are limited to the number of tags that we can afford to purchase. So MoveBank is helping us sort of overcome this a little bit. The other great thing about MoveBank is that to upload your data onto MoveBank, you need to undergo a bit of a process of outlier removal and formatting in a standardized way, so that any data that you collect from MoveBank is already in this standard format, and you can use the MoveBank tools that are available on this website pretty much straight away. There's also a number of R packages that are written assuming MoveBank naming and formatting conventions, and we'll be using one of those today. So just to give you an idea of what this looks like, if we go here to this data tab and map, it should bring us up a global map with spots that indicate all of the places where studies have been conducted and the data is now up on MoveBank. It might take a little bit of time to load. So there we go. So in gray, we've got all of the studies, and in green are the studies where people have specified that they want their data to be viewable by public users of MoveBank. And we can go down here, there's like a list of all of these studies. And if we select one and look at it, it's just a little slow. Then we'll be able to see the tracks that study collected, and we can have a look at the information about this study. So this was a study that was conducted by Rita Patricio and others, where they tracked adult female green turtles from nesting beaches. And they give us a little bit of information about when this was conducted, how many animals they tracked. You might even have metadata on the animals' biology or reproductive state or whatever on here. We also have a number of studies where the authors have allowed people to download their data. So if we look at those, I had a little search and I picked out this data set. So if you filter by study name and you type in albatross, you'll get this search result here on the top left. And I picked out this data set of grey-headed albatross that was collected in New Zealand. And they tracked 24 animals using GPS. So what we can do is making sure first to check what the sharing agreement is for this study and the requirements that they request for proper acknowledgements and citation. We can then proceed to download their actual raw data. So if we go download the data, agree to the license terms, we'll be downloading a CSV file, and we want to also include UTM coordinates and a local timestamp time in our download. So we'll do that, download, and I saved my data to my desktop folder, but save it to an easily accessible place, maybe the same place that you've got your markdown file. And can I just get some thumbs up once everybody's downloaded that data set? All right, 1 or 2 people are there. Okay, so we've got the data. Happy? All right. I'll also just point out while I'm here, if you're interested in learning about using R to analyze animal movement data, there's a really nice paper by Ju and others which highlights all of the kind of predominant methods that people are using to do this in R and the packages that are available for that. So I've provided a link to that study here. And today, what we'll be using is the CTMM package, which is the continuous time movement modeling package. And we're going to be using it to look at outliers in the data, telemetry error, and autocorrelation, which I'll go through one by one. So first of all, we need to install the package CTMM. CTMM. So by running this, and we'll load in that package, and we'll also load in tidyverse. All right, just if anyone wants me to stop at any point, just shout out and I'm happy to do that. Okay, I set my working directory to where I have my downloaded data file, and I input the name of that file in my read CSV function here so that I can bring that into R and have a look at it. All right. Now, essentially what we've got is we've got a data frame with 90,000 rows, over 90,000 rows. And the kind of key information here, which is common for any animal tracking data set, is you'll want your positional information, which in this case is our longitude and our latitude. We'll also have our timestamp here, which is a combination of our date and our time. And we've got a unique identifier for every animal that we have tracked. In this case, it's called individual local identifier, and it's just an integer value. Also, when we did our export, we specified that we wanted a UTM northing and a easting, and we wanted a local time zone. All right. Other information that your tags might be collecting can include things like acceleration, elevation, or depth. So those can be useful for looking at 3D movement or behavioral inferences. We might have some metric of telemetry error or spatial error, the accuracy that the device will give us. And we might have some tag diagnostics, things like battery life or temperature. These can be pretty useful for inferring why your tag might be performing poorly or why it might have failed, and if possible, adjusting the settings. So for example, if you're working with solar powered tags and your voltage is low and you're collecting very, very frequent locations, you might want to reduce the frequency of those locations to make your battery last longer. So if we summarize the data that we've got here, we'll just make a little table of our albatross data frame and look at the total points we have for every individual. We'll see that we have 24 animals that were tracked with somewhere between 2,000 to 5,000 locations for each one of those. And remember, these are GPS locations. We're going to now convert that data into a telemetry object. So this function as telemetry works on move objects. So we're using dependency, the move package, to interface with MoveBank through R and create this telemetry object. Because our data was sourced from MoveBank, it's already going to be in the right format for doing this. Otherwise, we would need to ensure that whatever data we're bringing in is following the MoveBank naming conventions that I mentioned earlier. And those can be found at this link in case you want to do that without uploading your data to MoveBank first. So we're going to create a move object albatross using this as telemetry function on our data frame that we brought in from our CSV file. And what that looks like, well, first of all, it's going to give us a minimum sampling interval, so the minimum time interval between successive locations for each of these animals. And the output is going to be a list of 24 elements. Those are our 24 animals. And each of those elements in the list is a separate data frame for each individual. We can pull out one of these. Remember that our animals are named by integers, and the first one was called number 1. So we'll call animal 1 is going to be from our albatross move object, animal number 1, and we'll plot that animal's track. All right, so along the x-axis here, we've got our x coordinates and our y coordinates are along the y. So this is essentially a map of those locations. We've got some, just keeping an eye on the chat as I go, but it seems okay. All right, so it looks like this animal has performed an area restricted type of movement and then done a long loop, which could possibly have been a migration or something like that, considering it's an albatross, that's likely. We'll be looking at telemetry error now. So within any tracking technology, we have trade-offs between things like the size of the device and the battery life. For example, larger devices might be able to contain larger batteries, but you wouldn't be wanting to use a very heavy device on a small animal. There's also trade-offs between the cost of the device and things like the precision that might be available. So Argos tags, for example, estimate locations using Doppler shifts of Argos satellites. And the error in terms of location accuracy that you might get from an Argos tag is within hundreds to thousands of meters, while GPS tags are typically more expensive and have location errors of tens of meters, so much more spatially accurate. Location errors are heteroscedastic, so that means that they're not uniform across all of the locations. The variance changes in time, and it's influenced by things like satellite reception and the kinds of hardware and software in the device, and also the fixed rate. If we're talking about GPS tags, for example, if a tag was collecting a position every 60 seconds, it's likely to be much more accurate than if it was collecting a position every hour or so. If we have inaccuracy, horizontal error in our tracking data, this is something that's a very important consideration, and it's something that is largely ignored in much of the existing analysis tools for animal movement data. The thing is that a location, a longitude and a latitude, doesn't necessarily mean that the animal was exactly where that record falls. If we assumed it was, it would be affecting the kinds of conclusions we could make about what habitat the animal was in, what distances it was traveling, and the home range size. So it is an important consideration, especially when the scale of error is relative to the scale of the movements of the animals. In our albatross data, we're working with GPS, so the accuracy is likely to be within tens of meters, and our animal is traveling 1,200 kilometers. So in this case, telemetry error is unlikely to be a hugely important thing to think about, depending on what our question is going to be. The data set that we downloaded from MoveBank doesn't contain any fields on location error. In CTMM, we can still work around this and estimate our location errors if we follow a vignette that I've included here. And that process involves modeling location error while we fit a movement model to the data. But it's much less reliable than the recommended method that the package authors recommend, which is to use calibration data. So I just want to show you an example data set from the package, which is this wood turtle GPS tracking data set. So if we can run this first line, that will bring in the turtle data. And again, our turtle data is a list of 4 objects, much like our albatross data, but we have 2 of these objects which are calibration data. So that means that the authors have taken 2 of their devices, put them in a fixed location, and let the device collect data without moving for a set period of time. And this provides information on the kind of scale of errors that we would expect to get from the same types of tags that we attach to animals. So this first element in our turtle list is a calibration data set. If we look at what the column headings are for that data set, we've got our timestamp, which is T, and we've got our location information, X and Y, which we would expect to be roughly consistent given that the tag isn't moving. And we've got this other feature, HDOP. HDOP stands for horizontal dilution of precision. And this is a unitless and device-specific measure of the magnitude of heteroscedasticity from the tag. And it can vary depending on satellite reception, latitude, the effects of habitat, so things like canopy cover, etc. And it's not an actual quantile or a standard deviation of our errors. We can visualize what that HDOP looks like. So if we plot our calibration data, we can see that these locations are essentially centered around the same place, but rather than being points, it's better to consider them as circles with a radius of a certain size. And here the radius is up to 200 meters. So if this was an animal, rather than saying, oh yes, our animal is here in the center of these circles, realistically, the animal could be anywhere within that error circle. And that's a really important consideration when you're thinking about what this data actually is telling you. I just have a question in the chat. Let me see what it says. Do calibration errors also depend on location sometimes? What would be the optimal number of calibration points, usually in a study of animals like albatross that move large distance and have GPS trackers? As in, if you set up a calibration in the far north hemisphere, is the calibration error likely to be different from a location closer to the equator? Yeah, I don't know how to answer that question. Hang on, what would be the optimal number of calibration points? So usually they recommend that you set your calibration for at least a few days. I guess it depends on what your sampling interval is like. If you want to collect a location every day, you would want to have a minimum number of locations in your calibration data set. But if you were collecting very high frequency information, you would only need a day or less than a day to do that. In terms of that latitude question, I'm not sure. I'll have to look it up and get back to you. Sorry. Okay. let's move on. So, okay. Then the next part of this is to use this calibration data to give an estimate of error to our tracked animal locations. So we're using this function UERE fit and our calibration data, so the turtle list elements 1 and 2. And we're going to be calculating the user equivalent range error. So this can be thought of as an absolute error in meters. And the way this function works is slightly different depending on what your input of your device specific error metric is and what kinds of devices you're using. Yeah, essentially what it does though is it gives us an estimate of our scale of errors in meters. And here we've got 2 different classes of location. Classes might be things like how many satellites were used to calculate a position. And so the error for each of these is going to be different depending on that variable as well. And then we'll be applying that user estimated range error to our tracks using the UERE function again. Okay. The next thing I wanted to touch on is outliers. So outliers are observations in the data that are beyond what we would expect based on the location error or the movement capacity of the animal. For a wood turtle, it would be really unusual to see it move 3,000 kilometers in an hour, for example. And so we can try and identify these outliers using this outlier function in CTMM. And this is based on a classification of maximum likelihood distance of speed, distance and speed. So with our albatross, we can plot that animal's outliers, and any unusually high speeds are going to be highlighted in blue and unusual distances are going to be highlighted in red once it runs. Okay. So, okay, so here we've got some unusual speeds and some far distances that have been highlighted by the outlier function. Now here, I would take this with a bit of a grain of salt because it looks like this track, rather than having a spurious random location in a strange position, is quite consistent. And this would be a plausible movement path that animal could follow given its movement capacity. So I wouldn't be concerned in this case about removing any of these locations. One thing is though that CTMM doesn't currently have the models built in to deal with things like migration and drift. And so essentially if we wanted to do anything further with this albatross data set, what we could do is we could cut out this migration pathway and just look at this kind of central, highly intensive space that was used. That's one way that we could do it. And the developers are currently working on building some of those models for other things. But what I'll do is I'll just continue looking at the turtle data from now on. So if I also plotted another animal from the albatross just to have a look at what that movement pattern was. And it seems like this migration pattern is quite consistent throughout the data set, which is cool. All right. So if we look at our turtle data, we have our calibration data we could run this outlier function on. And there's a couple of points that come out as potentially being problematic in the calibration data. And in the turtle data, there's one really obvious point that sort of seems to be a speed and distance outlier. If we wanted to, we could remove this point. The way that I wrote in here to remove it would be to look at, because it's come out as being an unusually large speed, we could filter out the point by a minimum speed threshold. So I've just applied a label bad for a speed exceeding a minimum amount, and then subsetting that turtle 3 to exclude that bad point. So if we run this, then we rerun the outlier function and that point is removed. We have a few other points that come out as potentially being problematic, but I don't think there's anything too concerning here. Everything seems to be within the kind of scale of each other. And there's also a another plot that we can look at, plot out, which shows us that all of these locations in terms of their deviation and their speed are within sort of the confidence intervals of each other. So there's nothing really too concerning here. Okay, the next thing I wanted to talk about is variograms. Variograms are a way that we can visualize autocorrelation structures in the tracking data. Now, what do I mean by autocorrelation? If we have an animal that was at a location at time 1, at time 2, it's more likely to be close to that first location than it is in 3 months from now. So autocorrelation essentially says that points that are close together in time are more likely to be similar than points that are far apart in time. And the same principle applies to the velocity that an animal is traveling at. So if an animal is moving very fast at time point 1, it's likely to be moving very fast at time point 2, whereas in 3 weeks, it might not be moving fast anymore. So we have a phenomenon of not only positional, but also velocity autocorrelation in the data. So the blue line, the blue line indicates large speeds. So faster movement than we would expect given, sorry, I'm just answering a question in the chat. Yeah, faster speed than we would expect given the error and the movement capacity of that animal. Okay, so yes, all right. And we're going to be using the turtles to look at this variogram method, look at the autocorrelation structure of our turtle data. And this uses this function variogram will produce a plot. So if we just run this whole chunk, we'll get a plot that looks like this. Essentially what we have along the X is the time lag, and along the Y is an average square distance that the animal traveled. Now, to interpret this plot in terms of autocorrelation, what we're looking at is where the curve seems to flatten out. And here the curve is flattening out at around 20 days. What that tells us is that if we wanted to consider points in our data set as being independent of each other, they would have to be separated by at least 20 days. And this is not the case a lot of the time in our tracking data. And assuming that those points are independent would be what we would require for underlying assumptions of a lot of different models that we apply to tracking data. Things like kernel density estimates, species distribution models, and any type of thing that requires independence as an assumption of your model. So without considering autocorrelation, you'll be violating your model assumptions in this case. The other thing to notice here is that this flattening out happens at around 2 to 3 hectares. So that's kind of the scale at which we would expect a home range to be, in terms of area. Yes, okay. That time lag as well is equivalent to the amount of time it takes for an animal to cross that home range several times. All right. The kind of crux and most powerful thing about CTMM is that we are using all of these considerations to fit hypothetical movement models to our tracking data, and that this movement is treated as a continuous time process rather than a discrete process. And this is these kinds of models are derived from principles in geostatistics and particle physics. and they take into account autocorrelation of positions, autocorrelation of velocity, and this range residency, so the tendency for an animal to remain in a restricted area. Whether or not your tracks adhere to each of these three things is what will be used to classify what kind of continuous time stochastic process model we choose. More details on how to do this can be found in the CTMM package documentation and in this paper that I've included here. So please have a look at that if you're interested in some more details. We have several candidate models that are possible fits for our animals, and which one we choose, as I mentioned, depends upon whether or not they are autocorrelated in position, autocorrelated in velocity, and whether they adhere to range residency. So the first one is called identically and independently distributed models. It assumes that our data are uncorrelated in both position and velocity. We've also got Brownian motion, which assumes that the movement is regularly diffusive, so the animal keeps getting further and further away from the first initial location. In this type of model, velocities are not autocorrelated and space use isn't limited to a restricted home range. We've also got our Ornstein-Uhlenbeck models, which combines this Brownian motion, this diffusive movement, and a tendency to remain in a restricted area. We've got integrated Ornstein-Uhlenbeck, which is like the Brownian motion diffusive model with velocity autocorrelation, but without restricted space use. And then we've got the Ornstein-Uhlenbeck foraging model, which encompasses both autocorrelation of position, autocorrelation of velocities, and restricted space use. Okay, there are different ways that we could go about selecting a model. I just want to run quickly through the kind of automated process of doing that in CTMM. So we use a CTMM guess function, which gives sort of an estimate of the model input parameters. When we run this, I want you to just copy it and run it in the console, because we'll get some interactive sliders that will help us set our model parameters. So this here is our semi-variogram that we made earlier, and below it is the model based on these guessed parameters. And if we go to the toggle here, we can adjust these. The problem here is that it looks like that that's flattening of the curve seems to really be underestimated. So we need to increase the asymptotic variance, so the variance of the asymptote where we get that flattening of the curve. And we can increase that until we feel like it encompasses a good position. The other thing is here, the positional autocorrelation timescale, so the point in time in which the curve flattens out, seems to also be underestimated. It's saying that we reach that asymptote much sooner than is indicated by the data. So we'll have to adjust that up as well. I'm going to have a drink of water. Okay. And so we can play around with these parameters a little bit until we're comfortable with our model fit, something like this, maybe. And then we'll be saving this to guess. So this is going to create an object in our environment that is called guess. And it includes those input parameters for our model fits. The next function that we'll run is CTMM select. What this does is it considers these parameter estimates that we've input, and it iterates through all of the candidate models and selects the best one based on AIC. This line takes a little bit of time to run. So if you just run that, and then while it's going, I want to quickly show you the Shiny app for CTMM. So if we go to, so CTMM has a Shiny app, which you can find, I'll send a link through at the bottom of this markdown as well. It basically follows the same workflow as what we've been doing in R today. We have some information here on vignettes for using the package and video demonstrations of this as well. We can import our data from MoveBank or your own data as well, or you can use data sets from the CTMM package like we today used the wood turtle data. And then we can load this data up. And it shows us, much like in R, we have 4 elements. We've got our 2 calibrated data sets and our 2 turtles that were actually tracked. We have a start and end time for each of those tracks, the sampling intervals, so how frequently we were collecting locations, the duration of those tracks, and the number of points that were calculated, or that were recorded, I should say. We also have some nice little automatic plots that we can generate. So this one is all of those elements combined, but we could look specifically at an individual if we were interested in just one animal. This one, faceted plot of just that individual, and that outlier that we removed is still there in this plot. We can also do our error calibration directly in here and assign errors to our locations. There's also the tools for filtering outliers here, subsetting in time. If you were interested in just a particular time range for your animal, you could do that here. And also for looking at our variograms and our model selection, that's here too. And some of the other subsequent analyses that you can do in CTMM, looking at home ranges, overlap between animals or timestamps of a single animal, occurrence distribution, and estimating speeds and distances based on these continuous time movement models that we've applied to the track. Okay, how is everybody going with their CTMM fits? Are there anybody finished running theirs yet? Thumbs up or still running? That's all right. It takes a little while. While it's going, I can show you, because mine ran before, so I'll show you what the output of that will be like. What we get if we run a summary of that fit object that we're making is parameter estimates. So we'll get an estimate. Well, first of all, it will have told us what is the best selected model based on all of the models that it ran. And for us, it was the Ornstein-Uhlenbeck. So remember, we have positional autocorrelation and a tendency to remain in a home range. And we'll have an estimate here of the effective sample size. This is essentially proportional to the number of range crossings, so the number of times an animal crosses its home range throughout the tracking period. We get an estimate of the home range area, and here we might get an estimate of speed, but because our model doesn't consider velocity autocorrelation, we won't get that in this case. And we'll have estimates for each of these and confidence intervals, as well as estimates of the error for our different location classes. Okay. Yes. So that's essentially what the output of that function does. We can also check what that model fit would look like by plotting our semi-variogram with our fit object on the top of it. And when I ran this, didn't work. When I ran this earlier, my model was still not looking like it was doing a good job of fitting that tracking data. So I still need to play a little bit with my input values of my autocorrelation timescale and my asymptotic variance to create a model that fits nicely. What was that in the chat? Someone wrote. So OU is not the default model. It is the model that was selected. So when I run this, all of the models that I kind of went through before are run, and OU is selected as the best based on the AIC. So it's not the default. All right. Okay. All right, so now that we've done that, just for fun, I included a little function here for calculating the home range. The default way of calculating home ranges in CTMM is by using an autocorrelated kernel density estimate. A kernel density estimate is a pretty commonly used way of estimating home ranges or spatial distributions. However, as I mentioned before, the kernels don't take into consideration autocorrelation. So this AKDE is kind of at the forefront at the moment of being the most accurate home range estimator. And the other thing about it is that it can provide us with confidence intervals, which I'm not aware of other space use estimators that do the same thing. So that's why I favor this AKDE. So we can run one for our turtle using our fitted model. Now remember that our fitted model was not so beautiful, but we could use a nicer model once we've gotten the right parameter adjustments. And that will give us this, I didn't really need to run that, did I? So that will give us this contour, this nice little squiggly blob, where the dark line here is the 95% utilization distribution, or essentially the area in which the animal was 95% of the time. And the gray lines are those confidence bands around that area. Basically, that's as much as I wanted to show you. I just wanted to reiterate the take-home messages that CTMM can help us account for telemetry error, autocorrelation, and irregular sampling frequencies in our animal tracking data. And these are key considerations when we're thinking about subsequent analyses and the inferences that we can make from them. The vignettes for some of the things that we worked through today, including AKDE, are really useful and easy to follow, and you can follow the links that I've provided in this markdown. Also check out the web app and the CTMM initiative webpage for more resources. And the last thing I'll just say quickly before I finish is, for those of you that aren't aware, we have a little student group called Rome. We are researchers of animal movement, mostly based at JCU, but we also have members who are interstate or at other universities, and we meet fortnightly and talk about concepts and techniques for dealing with animal movement data and nerd out a little bit. So if you're interested, don't hesitate to contact me and I can give you some more information about Room and the things that we're working on at the moment. Are there any questions?
Can you hear me?
Yep.
I just have a question about in your script, the part that talks about how the model is slightly biased downwards and underestimating diffusion. And you said it was because of the continuous velocity at short time lengths that the model is not accounting for. What would be like your solution to that? What would be the next modeling step to better account for the downward autocorrelation? Is it?
Yeah, well, the problem with this model is that it's likely that, so sometimes we get an OU model selected because of an artifact of the sampling regime of that data. So for example, if we weren't collecting data at fine enough time points to model the autocorrelation of velocity or something like that. So at at this point, I think what I would do is I would have to set a parameter estimate, like a larger parameter estimate of sigma, that autocorrelation variance, asymptotic variance parameter, in order to account for that. But I also think that part of the reason why that's happening in this track is if you look at the semi-variogram for the whole thing, it reaches that asymptote and then it drops down again. So I think that that's pulling that parameter estimate down, and possibly it's possible that that track isn't adhering to that home range behavior after a certain timestamp. So you might be getting some drift later on in the track, in which case you could ignore that and just fit a model for the first part of the track that does fit that pattern nicely. Because that's also one of the assumptions of these models, is that they have to meet that range residency pattern. Yeah.
Cool. It's like a whole different world. Seeing this animal tracking data, just very different from everything I do and probably others do. So it's really cool. I'm sure you and Kai are like experts, and you know this like the back of your hand now. But it's all yeah, super cool. The other question I had, if anyone doesn't have any other questions, was like, what would be like the biological reasons for like a diffusion model versus OU, which is more home ranges? So that one I can understand. But like, what would be like a diffusion model example versus the other more complicated models? What would be like the biology of the system?
It could be, I mean, it could be an animal that's like a nomadic animal, for example. So if you had an animal that, rather than, you know, having its home resting spot or its home foraging area or something, an animal that just moves, drifts, that might be the reason why. Yeah. Or the other thing is you might end up selecting a model like that because your tracking duration isn't long enough to allow the animal to cross that home range several times. So the authors of this package, they talk about that number of range crossings as like your effective sample size. So if your effective sample size is very low, you might not be able to model that home ranging behavior, even if it is occurring as well.
Yeah, I was just thinking, I used to work on ground squirrels, and basically two things happen to newborn ground squirrels. If they're a male, they disperse elsewhere. They'd always leave the colony somewhere, and they don't stick around. The females always stay around though, and they inherit a part of their, it's like a matrilineal lineage. So they basically inherit part of their mother's land, essentially. So I was just wondering if the dispersal part would be, like the diffusion part would be good for like the male squirrels, because they're like going kind of randomly into the environment around them.
I don't know, possibly. Yeah, that's kind of one of the limitations of this package at the moment as well, is that it does all this great stuff, assuming that your animal is a range resident. But any other, what we call like movement syndromes, which are different things like diffusive behavior or migratory behavior or nomadism, they're not really dealt with yet. Yeah. Can it account for geological barriers? So at the moment, not that I'm aware of. I think the package developers were saying that what you could do is build a model that allows diffusion, but conditionally in some directions, and then account for your geological barrier that way. But it's not actually built into the package yet to do things that way. The way that I've seen some people, if you're looking at things like home ranges, for example, some people, they just calculate their home range without considering barrier effects, and then they like plot the barrier on top of the home range that they do, which is not, you know, statistically quite right, but that's how people get around that at the moment. Yeah.
Awesome. Any other questions for Emily before the expert leaves Kodar for a bit? I'm sure they can email you, right? If anyone has any questions.
Yeah, if you have any questions, you can email me. Yeah, that's fine.
Super cool. All right, going once, going twice. All right, thanks so much again, Emily, for doing this. This is awesome. I'm sorry, I forgot to give you a proper intro at the start. I'm just distracted, but yeah, it's been wonderful hearing about this like new world. Usually our talks are either like super genetics focused or super like stats focused, or something just about plotting. But it's so nice to have it's like this other world of data that I never even knew about until now. So it's wonderful. Yeah.
Thank you. Thanks.
And 3D home ranges. Wow. Pretty intense.
Yeah, that would be crazy.
All right.
Well,
thanks so much,
like
everyone.
Birds and all the normal animals, fish, all have 3D animals, all the 2D turtles, or things that satellites can only track animals on the surface. So, yeah. Thanks, Emily.
I think a lot of the time that's like a limitation of these tracking devices, right? Very few tracking devices are collecting 3D information that is accurate enough to model that kind of thing. And yeah, I've rarely seen people look at things on 3 dimensions. Like usually when someone is reporting on dive profiles or flight paths, it's like vertical dimension only. So it's cool to know that they're developing a 3D home range. That's interesting. I wonder what that will look like.
I know for my friend.
Well, that's kind of why I asked about the geological barriers. Go ahead, Kevin. What are geological barriers on terrestrial animals? If there's a hill, you can obviously say if it's actually there and the air radius is quite small, it's on that mountainside somewhere. But if you had like an altimeter data or something as well, you don't need the reception off of the satellite because they might be inhibited by the actual mountain range. So you can have like an altimeter value.
Yeah, there are things that you can do with like cost distances and step selection functions not in CTMM, but you can like input a difficulty, like a raster that your animal moves across with difficulty values based on things like that, like how possible it is that they're going to be able to access that pixel, and build models that way. But that's like a different type of analysis altogether.
Awesome. Thanks so much. Anyway, we're pretty much out of time. So if you have any more questions, feel free to email Emily. And thanks again, Emily.
No worries.
Next week, we don't have anything planned just yet. I think we're